So, the presentation that I want to give you today, I'm going to divide into 4 parts. First, I'm going to give you some background on the use of porphyrins as a biological indicator of mercury exposure. Then I'm going to talk about the specific issue of genetic toxicity associated with a polymorphism that affects porphyrin metabolism and heme synthesis. Then I want to talk about some other polymorphisms that we have found, common polymorphisms in the general population that increase the risk of mercury neurotoxicity. And then I want to give you a brief overview of some of the studies that we've been doing in children. This story began over 30 years ago now when colleagues, in particular Bruce Fowler and myself at the National Institute of Environmental Health Sciences, hypothesized that like lead, mercury, and other thiol-directed metals should be able to modify the heme biosynthetic process, and in particular porphyrin metabolism, in such a way as to serve as what we called then a biological indicator, and which we later used the term biomarker for metal exposure. And here are a couple of the early papers that we published on that. In fact, Mercury in 1977, a year later, comparable effects that we observed with regard to arsenic. I want to give you some background on the biochemical basis of this effect. This is the heme biosynthetic pathway. This is just a single process that extends from here to here. And this is a process I want to emphasize that goes on in every single cell in the body. This is not something that is restricted to erythropoietic tissues, because heme is a requirement for a large variety of cell functions, not just for hemoglobin in terms of transporting oxygen and carbon dioxide back and forth from the tissues, but heme is also necessary for intermediary metabolism, for cellular energy production. It's a component of cytochrome oxidase, which is part of the electron transport chain, and oxidative phosphorylation. And it's also required for, in the central nervous system, neurotransmitter synthesis. Regulatory heme is now known to be an essential constituent in the construction of neurotransmitters and neurotransmitter receptors. This is a process that starts out with just 2 amino acids, glycine and succinic acid, both from intermediary metabolism. And from those 2 amino acids, we end up with all of these porphyrins and ultimately heme. The first step involves just the synthesis of this, combining these 2 amino acids to aminolevulinic acid. And then we go on to form this pyrrole. It's called porphobilinogen. And then 4 of these combine to form the first of the porphyrins, which is uroporphyrin. The reduced form is uroporphyrinogen. It has these reduced nitrogens in the nucleus, which makes it reduced. The only distinction between all these porphyrins is the number of carboxyl groups on these outside rings. Here they are over here on porphobilinogen. You can see what they are: acetic acid and propionic acid. And the first porphyrin in this ring has 8 of these, so it's 8 carboxyl porphyrin. It's very soluble in the urine, and so it was dubbed uroporphyrin. And this uroporphyrin is just sequentially decarboxylated on down here to coproporphyrin by the same enzyme, number 5, which is uroporphyrinogen decarboxylase. And again, these porphyrins just differ from each other by the number of carboxyl groups. This one has 8. This one has 7, where this acetic acid, this group, is replaced by a methyl group, and then 2 methyl groups, and so forth, on down to coproporphyrin. It's called coproporphyrin because it's not very it's not very water soluble, and would prefer mostly to be excreted in the feces. Copro is the Greek word for feces, so basically that's where it was predominantly found to begin with, but a lot of it still gets excreted in the urine. All of these 1, 2, 3, 4, 5 porphyrins appear predominantly also in the urine. Then coproporphyrin gets oxidized by another enzyme called coproporphyrinogen oxidase. That's number 6, to protoporphyrin, and then it gets reduced and iron gets incorporated, and we have heme. Now these two enzymes, uroporphyrinogen decarboxylase and coproporphyrinogen oxidase, have sulfur constituents, which are part of either their active sites or their protein structures. And irrespective of where those sulfur groups are, maintaining those sulfur groups are very important to their enzymatic activity. And so as it turns out, mercury, we found, specifically inhibits at these two sites in the heme biosynthetic process. It doesn't totally inhibit unless you have an awful lot of mercury, but it does certainly impair the progression of these porphyrins through the process and affects heme biosynthesis. And so mercury inhibits the step specifically of uroporphyrinogen decarboxylase that converts the 5-carboxylporphyrin to the 4, and then the next step that converts the 4 to the 2-carboxylporphyrin. Now typically all of these porphyrins are formed in excess of what's needed for heme biosynthesis, and so the excess typically get excreted in the urine in a pretty well established pattern. And when mercury is present, these two porphyrins in particular tend to accumulate and are present in much higher amounts. So if we separate these porphyrins from urine, and using HPLC, this is a technique that we developed about that long time ago, and to separate these porphyrins. And you can see very distinctly there's uroporphyrin in the normal urine of an unexposed. These are actually animal studies. Doesn't matter what species you use, it all looks the same, including humans. The uroporphyrin is always present in kind of moderate amounts, as indicated by the size of this peak. Then there's always very little amounts of porphyrins with 7, 6, and 5 carboxyl groups. And then coproporphyrin, even though it's more lipid soluble and would prefer fecal excretion, appears in much higher amounts in the urine than the other 4 combined. And this meso is just a synthetic porphyrin that we use as an internal standard. No protoporphyrin ever appears in the urine. It's much too lipid soluble. Okay, in the mercury profile, we have quite a different picture here. Because of where mercury inhibits the conversion of these porphyrins onto heme, we get a big increase in the pentacarboxylporphyrin, and we get a big increase in the coproporphyrin. And in addition to that, we see the appearance of this intermediate porphyrin that comes out on HPLC just before coproporphyrin does. And we didn't know what this was to begin with, so we dubbed it pre-coproporphyrins because of its appearance just prior to coproporphyrin on the HPLC profile. Well, we were, of course, when we first saw this, very interested in this pre-coproporphyrin because as toxicologists, we thought this might serve as a specific biomarker of mercury exposure. Nothing else causes this to occur, and we never saw it mentioned in the literature before. So we did a lot of work in terms of characterizing this pre-coproporphyrin. We exposed rats to, in this case, methylmercury for about 2 months, and then we isolated their urine, and we isolated this material from the urine right down to its crystalline form. We subjected it to mass spectrometry, and we found that it had a mass that was comparable to this chemical. We've been in contact with folks over in England who had been working on these so-called isocoproporphyrins for a long time, and one of them had suggested it probably was this material. And what this porphyrin is actually pentacarboxylporphyrin with the propionic acid group that normally would go here removed in exchange for this methyl keto group. So we have 1, 2, 3, 4, and then there would have been 5 propionic acid groups to make this pentacarboxylporphyrin, but instead we now have 4 carboxyl groups on here, so it's not pen anymore. It's a coproporphyrin, isomer of coproporphyrin, or isocoproporphyrin, with a keto group attached to it. Well, we were very interested in knowing what the etiology of this was and how specific it might be to mercury, so we did quite a few studies on this in animals. And what we think is happening is shown on this slide. Actually, this heme ought to be over here. It's kind of got flipped. Just make sure this is, nothing more is distorted here. So here's the part of the heme pathway that we are referring to here. This is 5-carboxylporphyrin. Biuroporphyrinogen decarboxylase normally goes to coproporphyrin. And then coproporphyrin normally goes to 2-carboxylporphyrin and then on to heme, which should be out here. Okay, in the presence of mercury, the uroporphyrinogen step is inhibited. The 5-carboxylporphyrin accumulates, and as it does, it will compete with coproporphyrin as a substrate for the next enzyme down the chain, which is coproporphyrinogen oxidase. Now, instead of all of the material just going straight to 4-carboxylporphyrin and out to heme, it's now used by this enzyme to produce a compound which is called dehydroisocoproporphyrin, which is then further oxidized in the cells to the KICP that we see. So just looking back at the heme pathway, this porphyrin accumulates, typically would go from here to here in a linear fashion. Instead, when mercury is present, this accumulates and competes with this porphyrin as a substrate for this enzyme. So this is going to be in competition for this enzyme as a substrate, which doesn't normally happen, or does very minimally. And when this is oxidized by this enzyme, then we get that atypical porphyrin formed. Well, we did an awful lot of studies in animals to demonstrate the efficacy of this urinary porphyrin change as a biomarker of mercury exposure, and I don't have time to go into all those individual experiments right now, but I just want to conclude this portion by describing how the porphyrin change serves as a specific biomarker of mercury exposure. So first of all, it certainly does, it reflects a biological response. And in terms of biomarkers, this is vastly superior to just measuring mercury in tissues. You can measure mercury in hair, blood, or urine, or whatever, but some forms of mercury will not even appear in those tissues, like methylmercury typically won't appear in the urine, or elemental mercury isn't excreted in the hair, so you're really not going to get much of a measure of it. A biological response actually reflects the amount of metal that is in the tissue, which is much more important in terms of determining its toxicity. So a biological response is what we have in the way of this porphyrin excretion pattern to mercury. We can also see this as an initial indicator of mercury exposure before any toxic responses occur. I'll show you later just how sensitive these porphyrins are as a biomarker to mercury. All you need is a little bit in the tissue, and you'll see incipient increases, at least in the porphyrin excretion pattern. That will continue to increase in a dose-response way with duration and with dose and duration of mercury exposure. We believe this is a specific biomarker of mercury exposure because we don't know of any other chemical or physical or metabolic condition that actually causes the specific increase in these three porphyrins that are affected by mercury. And it's pretty easily monitored. All you really need is a urine sample, so it's totally uninvasive. And if you have the analytical procedure set up, you can do a lot of these pretty quickly. Well, what about the application of all of this to humans? Well, we have for the past 15 or 20 years now been engaged in a validation study and going much beyond that In human subjects, looking at a big population that we have comprised largely from the Washington State Dental Association and the Washington State Dental Assistants Association. This is a population that was comprised, as I say, a long time ago in order to be able to validate porphyrins as a biomarker of mercury exposure. This is really a pretty old profile, but I still have it on here just to, it very clearly distinguishes that in a human subject with relatively low mercury exposure, we see a normal porphyrin excretion pattern, similar to what we saw in the animals. And over here we have a subject who had been working with mercury, preparation of amalgam fillings for some duration of time, and here his urine clearly reflects this profile change where you have a big increase in penta, which is just this little blip over here, huge increase in coproporphyrin, and here's the telltale pre-coproporphyrin peak, indicative of mercury exposure. Notably, we do have a little blip over here, so this can be part of some sort of a natural phenomenon where the pentacarboxylporphyrin might be used normally by the coproporphyrinogen oxidase to produce a little bit of this, excuse me, but not very much. We did a number of studies in collaboration with Vas Saposhin at the University of Arizona. He had access to dental exposed populations, mercury exposed populations in Mexico, and so we collaborated with him on a number of studies. And this was just a part of one of those studies where we were just comparing now for a number of individuals the porphyrin elevation comparing two groups of subjects with and without mercury exposure. And this was another part of a study where we were looking at dental professionals pre and post chelation. Here we see dentists, only 5 of them, but nonetheless they had about 20 micrograms per liter of mercury in their urine and excreted quite a lot of it, 12 to 16 times the original amount in the first 6 hours following DMPS administration. Same thing with the dental technicians, and even the non-dental controls who had mercury levels that were clearly within what we consider the so-called normal range of 0 to 5, excreted over 10 times as much during the first 6 hours following DMPS, indicative of how much mercury many of us might be carrying around unknowingly. And then in terms of the porphyrins, we see even during the first 6 hours, these porphyrins are all elevated in comparison with what we would normally see. And even during the first 6 hours, we see about a 50% drop in the urinary porphyrin concentration, consistent with the excretion of these porphyrins and the elimination of them from mostly the kidney, but other tissues as well that the DMPS is able to mediate. So this demonstrates pretty clearly how tightly connected biologically these porphyrins are to mercury in the tissues. And this is a little chart that I think just demonstrates or summarizes all of this relationship of urinary porphyrins to mercury body burden. On the y-axis here we have the relative urine concentration of either urinary mercury or urinary porphyrins and mercury body burden and time on the x-axis. And we see normally if you're not currently being exposed to mercury, even if you are, your body burden can be pretty high. That's the dotted line here, which you'd only know by doing a biopsy of the kidney or something and measuring the amount of mercury in it. Your urine mercury, however, if you're not currently exposed, can still be quite low. It's not an indicator. Urinary mercury is not a good indicator of exposure that has occurred some time ago. But if your body burden is high, your porphyrin level will be high. That's the beauty of the biomarker. It's in relation to the amount of mercury in the tissue, not the amount you necessarily see in the urine. If you give DMPS, Of course, the mercury comes right up, as we just saw, in the urine as it's being excreted. The mercury body burden declines proportionally, and so do the porphyrins. So the porphyrins are a good indicator of mercury body burden and will be in relationship to urinary mercury only as long as you are currently being exposed. Okay, well, that pretty much summarizes what I had planned to say with regard specifically to the biochemistry of the porphyrins and the porphyrin profile as an indicator of mercury exposure. Now I want to talk a bit about genetic susceptibility to mercury, and in particular in relationship to a polymorphism that affects porphyrin excretion. So in the human studies, typically, this is from a small subset of the 500 or so people that we were looking at in our validation studies from the dental professional cohort. And what we see here is just a typical dose-response relationship. Looking at the subjects within the general population of this cohort, we see this is the log of urinary mercury here. So 0 would refer to 1 microgram per liter. 1 is about 2.5. 7. This is about 7.5. Log 2 is about 7.5. 3 is 20, and so forth. So this is a range over which dental professionals might be commonly exposed. And we see that most people, about 85% of subjects, and it doesn't matter what population we look at. We looked at chloralkali production workers, other people with occupational exposure to mercury. About 85% are going to respond in terms of porphyrin excretion in a very predictable way with response to mercury exposure. But it always turns out, irrespective of what population we look at, there are always about 15% or so of subjects who just plain flat refuse to cooperate. Maybe we should call these politicians, I don't know. These are folks these are above the 95% confidence interval, and these people always respond with much higher levels of porphyrin, sometimes much, much higher, 10, 20 times higher amounts of porphyrins in their urine than would be predicted on the basis of the amount of mercury that they are exposed to. So how do we account for this? Well, one idea is that these people are just plain biologically different in some way from the rest of the population. And in fact, this idea is borne out. If we take a look at this panel on the right, we find that these people remain above this 95% confidence interval in terms of excreting very high levels of porphyrins, even after we remove the effect of mercury from the statistical analysis. This is a statistical procedure called residual analysis that is used to demonstrate that these folks, in fact, are true outliers. They're different from the rest of the population in some very specific way. That's why they respond differently. Well, how could they be different? Well, a very likely idea is that they're genetically different. And the most likely way they would be genetically different is that they have a polymorphism in a gene that encodes one of the enzymes that we know mercury affects when it modifies porphyrin excretion. So we went back to this entire cohort. There were over 500 subjects in this study, and we genotyped every single one of them. And we looked at the uroporphyrinogen decarboxylase gene to begin with. That's the first one that mercury inhibits to cause a big increase in pentacarboxyl porphyrin. And here we found no polymorphisms at all. There are 10 exons in this gene, which means that there are 10 areas within the entire gene where the protein is actually expressed. We looked at both exon as well as these intron-exon boundaries. All we saw in this gene was a polymorphism downstream of exon 1, and it did not seem to have any effect on, it did not seem to modify the effect of mercury on porphyrin excretion. So then we went on to take a look at the other gene, that's the copper porphyrinogen oxidase gene. And here we found two very prominent polymorphisms. This one in exon 5, which has turned out to be what's called a silent polymorphism, because even though there's a single nucleotide polymorphism, it encodes a change for the same amino acid, glutamine. Now it turns out that these are anything but silent. Basically, we didn't think this would make any difference if you just replace one glutamine for another in the enzyme. But in fact, these are now known to affect so-called epigenetic changes, where the folding of the protein and hence its eventual activity and stability and other physical characteristics might be significantly modified. We haven't really gone back and taken a closer look at that one, though, which we plan to do, because the other polymorphism in exon 4 encoded an asparagine to histidine substitution, which made a very significant change in the enzyme structure and, in fact, its activity. And this polymorphism, in fact, turned out to have a very substantial effect on the excess porphyrin excretion that we were looking at. So in going back, having found this polymorphism, then we went back to this population, and we asked, well, I mean, these people were excreting vastly higher amounts of porphyrins than would be predicted on the basis of the amount of mercury they are exposed to. How many of these people actually have that CPOX4 polymorphism? We called it CPOX4 because it was in the 4th exon of the CPOX gene. How many of these people actually have that? And we were quite surprised to see that of these people up here in the larger population, over 60% of these people carried that CPOX4 polymorphism. Now, this is quite a remarkable finding, because it's very unusual to find a polymorphism that can solely account for so much of a phenotypic variation in a human population. But over 60% of these people had that polymorphism. Well, then you ask, well, what about these people down here? How many of them had it? Well, as it turns out, if you look at these just underneath the upper boundary of the 95% confidence interval, only 38% of those people had that CPOX4 polymorphism. Going down here to the lower limit, only 16% of those people had that polymorphism. And down here below the lower limit of the 95% confidence interval, only 4%. So here's a bar graph that rates over 61% above the upper limit of the 95% confidence interval, and the declining prevalence of that polymorphism as you go through the confidence interval down below it. So these findings are pretty much in the expected direction and consistent with the idea that this very high porphyrin excretion rate that we are seeing in these people with mercury exposure represents the phenotypic expression of a CPOX4 polymorphism. Well, of course, we are very interested in knowing how CPOX4 might cause this very highly elevated pre-coproporphyrin in particular. And we think what's happening here is shown on this slide. This is a revision of the previous one, with heme again misplaced, ought to be out here. But nonetheless, what we think is happening is when mercury inhibits uroporphyrinogen decarboxylase and causes all this 5-carboxylporphyrin to accumulate, the CPOX4, which is a different enzyme from CPOX itself, will preferentially utilize the 5-carboxylporphyrin instead of the 4-carboxylporphyrin, causing an excess of this diiso and keto isocoproporphyrin to be formed. And those are the experiments that are currently in progress and seem to bear out this idea. This is an enzyme being biochemically different, has different kinetic properties from the parent enzyme, and will preferentially mediate this. And of course, the amount of CPOC or KICP that is formed directly depends on the amount of mercury that is present to inhibit this enzyme and cause the substrate for this enzyme to accumulate. So here again, we have a direct relationship between mercury exposure and an enzyme that will preferentially utilize this, producing the really excess porphyrin excretion that we see people with mercury exposure and CPOX4. Well, the next question that we wanted to ask is, I mean, all this is very nice. We have really good, tight description as to how a polymorphism in the copper-porphyrinogen oxidase gene affects porphyrin metabolism, but what does all this have to do with mercury toxicity? The principal concern that we have, of course, is with regard to the neurobehavioral effects of mercury, which are pretty much legend. And again, we went back and we, within the entire dental population in our cohort, they all underwent a very extensive neurobehavioral test. This is a test battery that was designed by my colleague Diana Echevarria, and it is probably the most extensive neurobehavioral test battery that is now available. It's all done on a touch screen, computerized and so forth, so there's really not too much subjectivity in it. People come in and they take a breathalyzer test to make sure they're sober, and then they sit down and they and after screening for various drugs and so forth, they take this test, which takes an hour or two, and many neurobehavioral domains and functions within these domains are measured. And consistent with reports from other investigators, we know that elemental mercury affects these various domains. So if you have mercury exposure, you're going to have deficits in attention and working memory and visual motor function and perception and manual coordination skills and so forth. These are all very well established and confirmed by the tests that we gave this dental population. Then we tested them for genotype, the same test scores we looked at with regard to genotype. So in comparison with subjects who had the wild type form of copper-porphyrinogen oxidase, we found that those with CPOX4 performed more poorly or less well on all these various tests that are listed here, and there are various tests within these domains as well. So under attention, there may be 3 or 4 separate tests that measure different aspects of attention and so forth. And these results have all been published as well. And we found that in addition to affecting these various neurobehavioral domains, there were additive effects associated with those who were also exposed to mercury. Not interactive effects, so there was no synergy here. But these are effects that are largely additive, as indicated by these pound signs or asterisks. So where you have a deficit in attention associated with CPOX4, you get the additive effect of mercury associated with that. And you can see that for most of these domains, there are additive effects associated. So what we think is going on here is that CPOX4, you remember at the beginning I mentioned to you that mercury is, heme is required for neurotransmitter formation in the central nervous system. If you have wild-type CPOX4, you have abundant heme, you have sufficient neurotransmitter production, and that means that these various neurobehavioral functions, behavior, cognition, so on and so forth, all of them are going to be operating on what we would consider a normal level. It would take quite a bit of either environmental or psychosocial stress in order to impair these functions. And in the case of mercury, the impact of mercury on these various functions is going to be more or less selective, depending upon whether you have some additional polymorphism that affects these functions, or on the dose and duration of mercury exposure. On the other hand, if you've got CPOX4, you've got impaired heme synthesis. We now know this. You're going to have deficient neurotransmitter function, and you're going to be operating at a lower threshold with regard to these various functions. So the effect of mercury on this, the same dose of mercury that has only a selective effect normally, is now going to have a much more profound effect. So what we say is that CPOX4 lowers the threshold of neurobehavioral domains that are impacted by mercury. So we conclude from all of these studies, most people, 85%, they will respond to mercury in a very predictable manner as far as porphyrin excretion is concerned. If you have the CPOX4 polymorphism, this accounts for the high excretion of the atypical porphyrin, and also for the other two porphyrins, and in addition for increased neurobehavioral toxicity among at least 15% of mercury-exposed subjects. We see the CPOX4 lowers the threshold for neurobehavioral domains that are also affected by mercury, and the CPOX, or rather the altered porphyrin excretion pattern, we view as serving as a metabolic biomarker of susceptibility to this form of mercury toxicity. In terms of the public health significance of this could be substantial, because here is the frequency distribution of CPOX4. The mutant is fully obvious or present in 2% of the population. Another 25% are homozygous for this polymorphism. So if we have 310 million people in the United States alone, at least 6 million people are going to be the full mutant homozygous for CPOX4. And so at least that many, if not 25%, are going to be at increased risk of mercury neurotoxicity associated with this polymorphism alone. CPOX4 is not alone as far as its ability to affect mercury neurotoxicity, or to lower the threshold as we describe it. There are quite a number of others that have been defined, and some we have already investigated. Here's 4 of them that we've looked at in pretty close detail or are currently looking at. Brain-derived neurotropic factor, the 5-hydroxytryptamine transporter, catechol-O-methyltransferase, and another one called tryptophan dioxygenase. Let me give you just a brief overview of what we have seen on a couple of these. So, brain-derived neurotropic factor, this is required for learning and memory. It is especially important during fetal development, as brain-derived neurotropic factor is now known to mediate the upregulation of gene transcription of those genes that are involved in neuron growth during fetal development. A polymorphism has been well described in the literature as associated with impairments in memory and attention, and people with this polymorphism are known to suffer a greatly increased prevalence of anxiety and depression, same as you see in people with mercury exposure. So the obvious question begged is, does this polymorphism increase susceptibility to mercury neurotoxicity? And here again, just looking at the same parameters that we know are associated with mercury, we went back to the dental population, And we observed that these folks, in fact, these are different domains than were affected by CPOX4, although some of them are very similar. You see a number of them are associated with memory and attention and so forth, what we know this polymorphism affects. And again, for virtually every single one of these, we have additive effects associated with mercury exposure. The higher the mercury, the more predominant these deficits in memory and attention and cognition and so forth becomes. You'll notice also that the prevalence of these polymorphisms are even somewhat higher than we saw with CPOX4. And so again, we have a considerable public health concern with regard to mercury exposure among people who carry this rather common polymorphism. Here's another one that we've looked at called the serotonin transporter polymorphism. It's called 5-HTT-LPR. Serotonin, as you know, is an extremely important neurotransmitter. It mediates especially mood and is associated with a number of other neurobehavioral functions. The transporter has an insertion-deletion polymorphism that is referred to as it's 5-HTT, which stands for hydroxytryptamine, which is another word for serotonin, 5-hydroxytryptamine transporter. This is called a long polymorphic region that is very amenable to polymorphisms in the promoter region of this gene that transports serotonin into its receptor. This polymorphism has been associated with anxiety and depression and suicidal behavior. Again, something that used to be prevalent among dentists who work with mercury for a long time, and now is certainly seen in the general population associated with this polymorphism. So again, the same question arises, and again, the same types of analyses demonstrate considerable conformity between what mercury does and what this polymorphism does, and how we have additive effects indicating that the threshold for the people with this polymorphism has been lowered, and so the presence of mercury is very likely to be much more profound among people with this polymorphism. And notice that the full polymorphism is present now in 18 to 19% of the U.S. population, and 39 to 57% of the heterozygous form. So a lot of people are potentially affected by this polymorphism and the additional consequences of mercury exposure. This is just a summary slide that shows these different polymorphisms that we've been looking at: CPOX, BDNF, serotonin, and catechol-O-methyltransferase is another one we're looking at. This one exacerbates the effects of mercury on confusion and coordination. This is another one of the high prevalence in the population. Been reported, literature is just full of reports on this. Of course, nobody but us has looked at the effects of mercury or other neurotoxic metals in combination with this polymorphism. So these are some of the conclusions that we have to date from some of these gene studies. These polymorphisms we see generally lower the threshold for the specific neurobehavioral functions impacted by mercury, which means that if you're totally devoid of a polymorphism, and I think that would be extremely rare for somebody not to have at least one of these, or in some other gene that we haven't even looked at yet, if you have one or more of these polymorphisms, you're likely to be at increased risk of the neurobehavioral effects of mercury, which is probably why we see so much concern associated with mercury exposure. What kinds of effects do these polymorphisms mediate most likely? Well, ADHD. What about autism? We hear a lot about that lately. No one has looked at any kind of gene-environment interactions in autism. I think it's incredible that this has not been looked at. People have essentially dismissed the notion that mercury could be involved in this, largely because of the meta-analyses that have been conducted on the thimerosal vaccine studies. But obviously, there are a lot of genes that have been identified. Autism is considered to be largely a genetic problem, but certainly environmental impacts have to be imposed on those genetic defects in order for this to be apparent. Depression, other pervasive developmental disorders, probably these gene-environment interactions associated with mercury and maybe other neurotoxic metals or environmental toxins could underlie quite a lot of this. And as I said, the high prevalence of many of these polymorphisms means that many, many people could be affected. Well, I want to conclude then with just a few comments on the studies that we have done in children. Many of you are probably aware that we were part of the dental amalgam clinical trial that was conducted to test the neurobehavioral effects of dental amalgam fillings in children. There were two of these clinical trials that were conducted from 1996 to 2006. And I apologize, these were perfectly well aligned when I looked at them this morning on my laptop, but they must have gotten flipped on this particular conversion to this laptop. Nonetheless, these are the baseline characteristics of the study population, which had about 500 kids who were randomly assigned to either an amalgam group or to a composite group. They were about 50-50 boys and girls. The age of the kids in both groups was age 8 to 12 at baseline, and they really had a lot of dental need. This study was conducted in Lisbon, Portugal. These countries do have socialized health care, but that does not include dental care. If you want dental care, you go to a private practitioner and pay for that out of your own pocket, which is typical of, I think, healthcare programs that are government in most countries around the world. So these kids had an average of 15 to 16 carious surfaces at age 8 when they were incorporated, or 8 to 12 when they were incorporated into this study. So they really had a very high need for this dental care. This study, by the way, was conducted by us at the University of Washington, but in collaboration with the very, very well-established University of Lisbon and the College of Dental Medicine there. These are the progression of urinary mercury levels in these kids during a period from baseline through 7 years of follow-up. The kids were given most of their treatment during the first and second years of the study, when they would come in and they would give a urine sample for mercury and porphyrin analysis, and they would take this very comprehensive neurobehavioral test battery. This was geared towards children. This was an internationally recognized test that was well accepted and suitably designed for children of this age. And we can see in the composite group, the urinary mercury levels didn't change significantly during the entire course of the study. On the right-hand side, we have creatinine adjusted. Younger kids have lower creatinine levels, so these are a little bit higher, and then they decline during the progression through the teenage years. But notice that urinary mercury from dental amalgam fillings does increase from up to about 2 to 3 times in the urine of these kids during the course of treatment, maximizing out at about 2 years after initiation of the study. This is the period of highest treatment, and treatment then was no longer necessary, or not as necessary, during the subsequent course of the study. So urinary mercury levels tend to drop off, but never ever does it get back to baseline levels. which indicates that, of course, there is some mercury being retained in the tissues. We were very interested. I certainly didn't expect to see that there would be porphyrin increases in the urine of these kids just due to the very low exposure to mercury that they were getting from dental amalgam. Nonetheless, we were surprised to see that in the porphyrins that are affected by mercury, that there were these non-significant but clearly evident incipient increases in the 3 porphyrins that were maximal during the first 3 or 4 years, first 3 years of the study, when the mercury treatment was the most prominent. And in fact, if we restricted these assessments to kids who are only 8 and 9 years old at baseline, in other words, smaller, therefore smaller kidneys, that would accumulate more mercury from the same dose that everybody else was getting. These porphyrin increases in these 3 porphyrins were even somewhat greater. Again, these are non-significant increases, but they are clearly evident, and they are consistent with the 3 porphyrins, and indicative, I think, of just exactly how sensitive these porphyrin changes are to mercury concentrations in the kidney, even at very low concentrations that derive from amalgam fillings. This is a slide from the JAMA paper of the results of this study that was published in 2006, and it just describes the results of the neurobehavioral tests that were given to all of these kids. In this case, there were 4 tests of memory, visual motor function, attention and concentration, and nerve conduction velocity. And these kids, within each one of these domains, there were, as in the case of our test battery with the dentist, there were anywhere from 2 to 6 different types of tests that were given. And basically what we're looking at here is a comparison of how the amalgam group performed on an annual basis compared with a composite group on each one of these tests, or the combined test scores, really, for these different domains. And the amalgam is closed circles, and the composite is the open circles. And we can see that there were no significant differences, really, between the performance of these groups with regard to any of these various tests. Well, of course, from my perspective, the point I just made was you really must go and determine whether or not there is genetic susceptibility. That was something that was not evaluated within this particular population when the study was done. So we went back to as many of these kids as we could track. By the end of this study, they were mostly graduating from school and were off to the 4 winds, but nonetheless, we were able to track down quite a few of them through the University of Lisbon and the team that had actually facilitated this study with us. We went back and we genotyped them for the genes that we were of concern in the adult dental studies. And here's some of the gene dose effects that these are very, very preliminary data and subject to further analysis and confirmation and interpretation. But the tests here that are highlighted in yellow are where we are seeing a gene dose effect for these various tests, where the wild type, we see very few negatives here. The more negative, the worse the performance of the kids in this gene group on these various tests. So for tests of, this is for CPOCs, so for tests of learning, memory, and total learning, we see a progressive deterioration of performance from wild type to heterozygous to fully mutant. We see that most of these tests of attention and concentration are progressing towards the negative. In the mutant form, the kids that had the fully mutant CPOX4. Similarly, a few here in visual motor function. This reaction time should not be highlighted. It should have been this other finger tapping test. But notice no change in nerve conduction velocity. So This is not a aberrant finding, so we would not expect any change in nerve conduction velocity associated with CPOX4. So similarly, we looked at one more I'm going to show you. This is the catechol-O-methyltransferase gene polymorphism, which is known to affect concentration and coordination functions. And here under memory, essentially all of these demonstrated a gene dose effect with the mutants forming fully mutant for the COMT polymorphism, showing worse performance than the heterozygotes or the wild type. Just a couple here under attention and concentration, and with regard to coordination, several under that showing a gene dose effect for loss of function in the kids who are fully mutant in that. So again, these are very preliminary findings and subject to reanalysis and interpretation, but I just wanted to show you this again to emphasize that looking at mercury alone in a whole heterogeneous population is probably not going to reveal what's really happening. It's really essential that we must focus on those who could be more susceptible because of various genetic causes. So in conclusion from these studies, we found urinary mercury is very highly correlated with the number of fillings and the time of placement in children. We found that while the urinary porphyrins did not increase significantly in these kids with the amalgams, given that they only had a couple of micrograms per liter of difference of mercury in the urine, still there could be substantial accumulation in the kidney where the mercury effect on porphyrin metabolism is predominantly mediated, and we were surprised to see an incipient effect here where the urinary porphyrins are sensitive to these incipient changes in the renal mercury content. And then finally, of course, we must conclude that genotype affects susceptibility to mercury toxicity in children as we had seen in animals, or rather in adults. So in terms of future directions, where we're going with all of this, or would like to go, there are a lot of genetic variants that have been published in the literature that are very well established as being associated with not only adverse neurobehavioral effects, rampant types of effects like depression or ADHD in both children and adults, but also with neurologic disorders like autism, other pervasive developmental disorders that we really need to investigate with regard to the interaction with mercury. What is the contribution of mercury to these disorders in people with these polymorphisms? We also want to examine relevant pathway-related genes. Looking at just a polymorphism is great, but how do we integrate that with the idea of neurotransmitter functions? We now have a so-called SNP chip, which has polymorphisms for 2,400 different genes on it, and that these genes are related to functions that are likely affected by mercury in terms of mediating neurobehavioral disorders like dopamine metabolism, metal transport, the whole heme porphyrin metabolic process, oxidative stress, and inflammatory processes as well. How does mercury mediate or add to or make worse these neurobehavioral functions when we can look at entire pathways? And then finally, we'd like to get to the point where we can contribute to the development of these gene profiles. It would be wonderful if you could send in a spit sample to 23andMe or something like that and come back with a result that demonstrated a very comprehensive type of susceptibility profile to mercury or some other environmental toxicant. We think that's a good direction in which to be striving. So that concludes my comments, and I will be happy to respond to any questions.
Will you use the mic if you're going to ask questions? Dr. Wood, as a clinician,
I have done the urinary porphyrin profiling with 3 different labs and sent in split samples to each of the labs to look at the results, and they're as disparate as you could imagine. How can, as a clinician, one determine where to get accurate information on porphyrin profiling?
Well, I don't know what labs you sent your samples to, and I do not know how therefore I don't know how those labs would be doing the porphyrin profiling.
Well, the labs were actually had very different collection techniques. One of the labs was Metametrics that used that recommended the collection be frozen and immediately and sent. Another one was the lab in Paris, and the other one was a LabCorp.
Yeah, well, as you point out, they all have different collection techniques and probably different processing procedures. And truth be known, I have no idea of how they actually analyze their porphyrins. Even though they all say they're using a method that we developed, none of them has ever contacted me personally about how to set up these procedures or how to interpret the findings. So if they're analyzing the same split sample and coming back with disparate responses, it's very hard for me to tell how to reconcile that. I can tell you if you sent the split sample to our lab and we analyze them under 2 different or 3 different occasions, you'd get the same results.
How close are we to feasibility of polymorphism testing?
I'm sorry, I can't hear.
How close are we to a feasible testing, genetic testing for our patients? How many years off do you think we are?
Well, you mean commercial testing?
Right.
Yeah, I think a lot of companies are setting up now to do this type of commercial testing. If you wanted to collect a buccal cell sample and just send it in for an individual polymorphism, I think these are, still kind of at the experimental stage. I don't know any clinical labs that are actually doing this for profit or for a medical diagnostic purpose. I think they're going to be really incorporated into these kinds of profiling that some companies are setting up, like I mentioned 23andMe, and there's quite a few of them around. But I wish I knew the answer to that. We have a genomics laboratory that does all of these profiling and polymorphism tests for us, but these are done specifically for research purposes. I think you need a microphone.
Here, I've got one. The children's amalgam trial studies were published in Journal of the American Medical Association and promptly used as the ultimate final justification for the use of amalgam by the dental community. How do you feel about that? Do you feel like because you're speaking to a room full of dentists who are at least highly suspicious of the safety claims for amalgam, if not more.
Well, I think I explained how I feel about it. I mean, my feeling about it was that the study was very substantive and very, I think, competently performed, but it went nowhere near far enough in terms of defining those who might be truly susceptible. And that's what we are attempting to do right now with this genetic polymorphism study. And I think when the results of these studies are published, they may tend to modify the conclusions that were drawn from the dental amalgam clinical trial substantially. But in the meantime, I think we have to recognize that that study did show that for the general population of children, and this was confirmed by the other study that was done at the same time by NERI, that there were no significant differences in neurobehavioral performance, a wide range of functional tests for children who received very heavy-duty amalgam treatment within the general aspect of the assessment of those studies. And I think the conclusion was that therefore amalgam should continue to be used, certainly where no other alternative is available, in order to provide dental care for those people who would not otherwise get it. And this is true like in Portugal, where our study was conducted, and it certainly is true in a lot of third world countries where dental care is non-existent. If there is any treatment available at all, it would certainly be with a dental amalgam, and where there would be a great deal of dental miscarriage if it were not provided. So, but I think, you know, in terms of the kind of work that we're doing here, we are able to demonstrate that, in fact, there are people who are genetically susceptible, and in fact, children may be even more so because of their immaturity of their developmental processes and the way they handle mercury and so forth, that you know, those results will ultimately modify the conclusions that were drawn from the dental amalgam clinical trials.
Dr. Woods, first, Rich Fisher, I first of all appreciate your coming and sharing, I think, a brilliant presentation with us. Do you expect when those final data analyses would be done on that CPOX4 segment of the population?
Well, for the adults, this has been completed, and those papers. You mean for
on
the
the
kids?
children.
For the kids?
Yeah.
Well, as quickly as I can motivate my erstwhile colleagues into taking this extremely large and unwieldy data set and addressing themselves to the types of analyses that we need to have done. I mean, once you get the data analyzed, then you have to sit down and figure out what it means. But since we've already done this for adults, that should be pretty straightforward. So I'm certainly hoping that you know, we'll have something ready for publication within the year, if not a lot sooner.
Great, thanks.
Yes. I've heard some comment that collection of specimens for urinary porphyrins would be best if the patient went to the lab and had it drawn. Can you comment on handling of the specimens?
Yeah. Yeah, I'll have to say, you know, urinary porphyrins, you know, a problem with them is that they're very labile. They're susceptible to, I mean, these, you saw what they look like, and they're big box-like structures, and they are light sensitive. If you take a container with porphyrins in it, and you let it sit out in this kind of light, or certainly light in the laboratory, you know, those will be deteriorated just sitting out in an open beaker, or urine, which is even worse. They'll be deteriorated within a matter of a few hours. They're very light sensitive. They're also sensitive to temperature. And the best way to collect porphyrins, at least the way we do it, is to have them collect a spot urine sample usually, first or second thing in the morning. And we immediately stick it in wrap it up. Usually it's done in an amber bottle. So they actually pee in a glass or a urine collection cup. We pour it into an amber bottle, which is completely light-proof, and then we immediately stick it in the freezer. Now, this is the ideal way to do it. And when we did all these studies in Portugal, they did it that way. I went over there and we worked with the technicians to show them exactly how to do it. And they did it that way. Actually, we had to split the sample up into a number of different fractions for mercury, which requires different handling, porphyrins, creatinine, and a number of enzymes that we were measuring too to assess renal function. But the porphyrin sample is immediately put in at least a -20 freezer. If it's going to be held for any prolonged period of time, more than a few months, it ought to be probably -80. But nonetheless, when it came time to ship these, we would collect them for when we had maybe 50 samples or more. They packed them in dry ice. They put them in a styrofoam box, and they shipped them for next day delivery to our lab in Seattle. And there are a few problems with that. It took us a while working with a customs service and so forth get them to understand what we were shipping, but nonetheless, it finally ended up working extremely well. I think we had to use DHL. We had a contract with them that this is how they would be handled. And but you know, and I say I don't know how these things are prepared shipping them to France. You know, if they're just in a vacutainer tube that is not frozen, you know, I'd be surprised if there'd be much left by the time they got there. But I have seen some of their analyses show very highly elevated porphyrin. So maybe they've got nitrogen or something in that tube that prevents porphyrin degradation. But they're very sensitive to light and to temperature, and they will not hold up under ambient light or temperature conditions. So they've got to be treated very carefully, and of course when they're being processed, they have to be treated very carefully as well. We have a special room that has no UV light in it at all. It's just a yellow light that we use, or a red light like you'd use when you're developing film, to handle these porphyrins, and they're all extracted colorimetrically very quickly, and then they are run through the HPLC very quickly as well. They're pretty stable once they are isolated from the urine, and they're in an acid solution, so that tends to preserve them pretty well. But I mean, I hope that answers your question. I mean, we've published these procedures in detail, including there's a publication called Current Methods in Toxicology.